The MassSQUIRM protocol aims to quantify lysine demethylase activity using isotopic methylation and MALDI mass spectrometry. First, perform enzyme-catalyzed demethylation reactions on peptides containing methylated lysine residues. Then, chemically methylate the peptide products of the demethylase reaction with deuterated formaldehyde to produce uniformly methylated peptides that ionize the same way by MALDI. Proceed to quantify each peptide species with MALDI-TOF mass spectrometry and account for isotopic overlap in order to identify relative amounts of mono, di, and trimethylated peptides. The MassSQUIRM results determine relative amounts of each demethylase reaction product based on changes in mass.
The primary advantage of this technique over existing methods like routine MALDI mass spectrometry is that MassSQUIRM accounts for differences in ionization between differently methylated peptides. We first had the idea for this approach when we developed a method to measure histone acetylation levels using isotopically heavy acetic anhydride. The challenge to this technique is extracting peak area data, which can be complicated by overlap of peptide isotopic envelopes.
For the demethylase reaction, combine 125 nanograms of recombinant lysine-specific demethylase 1 enzyme with 0.25 micrograms of biotinylated dimethylhistone H3 peptide in a final volume of 20 microliters demethylase buffer. Also, prepare a control sample containing 0.25 micrograms peptide without